All right, so in our last video, we talked about the action potential. The action potential is that wave of electrical activity going down the sarcolemma, the cell membrane of a muscle fiber or muscle cell. The whole point of the action potential in muscles is to cause muscular contraction. So today, in this video, what we're going to talk about is skeletal muscle contraction. So just to prompt some old information, right? Remember when we were talking about the organization of muscles? We said that like the whole muscle itself is bound by the epimysium, and that it is it contains bundles of bundles of bundles of bundles of bundles, keep going smaller and smaller and smaller, right? So the muscle itself is bound by the epimysium, and it contains bundles called fascicles, and each fascicle is bounded by the perimysium or mesium, and it contains like a bundle of muscle fibers. And each of those muscle fibers is basically kind of like an independent cell, but with really long and multiple nuclei, and it is bound by the endomysium, and it contains a whole bunch of bundles of contractile proteins called myofibrils. And that was about where we stopped. But now we're going to continue further. So these myofibrils, what you see here, this is a myofibril. Inside of the skeletal muscle fiber, you have a huge number of these myofibrils all gathered together. And the myofibrils are what makes up the contracting force of the cell. The more myofibrils you have in a cell, the stronger that muscle cell can be. These myofibrils are made of several components, but the most important components of them are what we call thick filaments and thin filaments. So if you look down here, so this is an arrangement of thick and thin filaments. And this is the way that the myofibril is arranged. So there's a few important things here. First, you have the Z-disc. The Z-disc is a line of proteins on a microscope. It looks like a ziggy-zaggy line. So that's why it's called the Z-disc. And sticking out from the Z-disc in either direction are thin filaments. This here, that is the thin filament. And then you have thin filaments sticking out in this direction and thin filaments sticking out in that direction. The thin filaments are made out of actin. Actin is a cytoskeletal microfilament, right? So it's a cytoskeletal component. In the middle here, we have the thick filament. So all of the thick filaments, just like all of the thin filaments, are held into place by the Z-disc. The thick filaments are all lined up and held into place, into alignment, by a set of proteins that make up what's called the M line that runs through the middle of them. This thick fiber is actually made out of a whole bunch of proteins called myosin, which means muscle protein because it's found so abundantly in muscle, though it's found in almost every cell in your body, it's found very abundantly in muscle. And all of these myosins are bound together and sort of twisted. And every myosin has a tail that sticks down this way and a head. And you can see all of these little knobby things. They look like golf clubs coming off of this thick filament. Those are the heads of the myosin. Myosin is a motor protein. What happens is that that head group can grab hold of something and it can pull. And this thick fiber is made of all of these myosins twisted together like fibers twisting to make a yarn. And these heads bind the actin of the thin fibers, thin filaments here, right? So the thick filament, that's what's going to do the pulling, and the thin filament, that's what's going to be pulled, and it's going to be pulled towards the M line. This whole structure, where you have a Z-disc, thin filaments, thick filament, thin filament, and another Z-disc, that is called a sarcomere. Remember, sarco means muscle. A myofibril is a polymer of sarcomeres, right? So in your myofibril, you just have sarcomere, 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 sarcomere. And the sarcomere is the base contractile unit of the muscle. The sarcomere is one contracting unit, and all of the sarcomeres acting together are what is going to contract the muscle cell. There are some important areas in this sarcomere, and I'm going to explain them using this image up here, but I want you to keep in mind this image down here, what things actually look like, right? So here we have our M line. M stands for middle. It runs down the middle of the sarcomere, and here we have our Z discs. They're ziggy-zaggy for Z, and they are what represents the end of the sarcomeres. The sarcomere is bound on either side by the Z discs. The sarcomere has an H zone, right? So this H zone here, the H zone represents the area where you just have thick filament. So here we see this is a stack of thick filament. There's no thin filaments there. So H zone, just thick, no thin. And it's called the H zone because if you look at, so this, and that, and this, that looks like an H. Well, it would look like an H if I could draw very well. But it basically looks like a whole bunch of H's together. There is the zone of overlap, and the zone of overlap is this area where you have both thick and thin filaments, right? So that makes sense that that would be called the zone of overlap. This whole area here, from say here over, oops, not all over that far, over to here. Let's erase that. From here over to here. That whole area where there's thick filament, whether or not it's overlapping with thin filament, that whole area is called the A band. And this area here, which actually spans two sarcomeres on either side of the Z disc, where you have thin filament but no thick filament, that is called the I band, because it looks like an I. And I know that an I and an H actually look about the same, but here's the way to remember them. So the composition of the filaments. Thick filament, we said, is made of a whole bunch of myosins. Myosin is the motor protein. This is a single myosin here. And as you can see, it's got this sort of golf club shape with a long, straight tail and this big knobby head. The head is the motor, right? And so the head is going to move that way. And it burns ATP to do that. The thick filament is made out of a whole bunch of myosin twisted together. So here you can see the actual filament part is all of these tails joined together, and you've got heads sticking out in every direction. And so they're arrayed in like a spiral around the thick filament, so you've got heads basically pointing every which way. In the middle of the thick filament, there's what's called a bare zone with no heads. That's where the tails start. So the tails actually start at the middle, at the M line, and so there's this area towards the middle of the thick filament that doesn't have any head groups, and then you have head groups going out from there. The thin filament is made of actin, specifically two intertwined strands of actin. That's these little red spheres here. And actin is a filamentous polymer that can be formed and it can be broken, but it is a large part of what makes up the cytoskeleton and cytocells. Here it's not being used for a cytoskeletal role, but it's still a strong fibrous protein. So actin subunits each have an active site that can bind to myosin. They're little like handholds that myosin can grab hold of. And that's what myosin is going to do. When myosin is prompted to work, it's going to grab hold of the actin at the handholds at the active site, and it's going to pull it in, and then it's going to reach down and grab the next handhold, and then it's going to pull that in, and then it's going to reach down and grab the next handhold, and it's going to pull that in. So the myosin has to be able to grab to these active sites, these handholds on actin, in order to pull it. There's two proteins that are super important that are associated with actin, and they are called tropomyosin and troponin. What tropomyosin does is it binds to the actin and it covers the active site. So when tropomyosin is bound to the actin, like it's you can't grab hold of it. It's slippery. So as long as tropomyosin is on actin, myosin can't grab hold. The handholds are not available. They're protected. That is tropomyosin. The next protein is troponin. That's these little snail-looking things here. All right? What troponin does is it can move tropomyosin. If tropomyosin is in place, then myosin can't bind the actin, you don't get any contraction. If troponin moves tropomyosin out of the way, it exposes the handholds. And then myosin can bind and you get contraction. So troponin is actually what's controlling whether or not you have contraction going on. It gets to say, yes, I'm going to reveal the handholds and we can contract, or no, I'm going to cover the handholds and we can't contract. And it follows a very simple chemical signal to do this. It binds calcium. When it binds calcium, it moves troponin or tropomyosin out of the way, and contraction occurs. When there is no calcium, it puts tropomyosin back into place, and contraction stops. And as soon as contraction stops, the muscle starts to relax. There are a few other important proteins that make up this system as well. So we've already talked about a few of these other features. So we've got the thin filaments, and we've got the thick filaments. Remember, thin filaments are made of actin. Thick filaments are made of myosin. The protein myomycin. Myomycin for M, that's what makes up the M line. That's the protein that's going to hold all of these thick filaments together. Next, we have titin. Titin is actually the largest single protein in the human body. There are protein conglomerates like collagen that are bigger than it, but collagen isn't just one protein. Collagen is actually a bunch of proteins stuck together. Titin is all one big giant protein that acts like a spring. And what it does is it runs through the thick, it attaches to the Z disc, and it runs through the thick filament. And so it's going to anchor this thick filament to the Z disc. And it also, much like a spring, is going to act to return things to the zero position. So if this gets stretched out, the titin is going to spring back and get everything to the zero position. And when this thing gets contracted in, and then suddenly you let go, right? You stop contracting the muscle, this titin is going to work like a spring that sort of pushes the M line away and allows the muscle to react. We also have nebulin. Nebulin is the protein that attaches the thin filament to the Z disc. Right? So it's going to hook the thin filament to the Z disc. These are all very important proteins. They're not nearly as abundant as thick and thin filaments, but they are what hold the thick and thin filaments together. One more important, and in this case very clinically important protein, is dystrophin. So right now, all we've been talking about is the contraction and relaxation of these myofibrils. Well, why would a myofibril contracting necessarily cause the entire cell to contract? In order for a contracting myofibril to cause the cell to contract, it would need to be hooked to the surface of the cell. And that's what dystrophin does. Dystrophin attaches to thin filaments on one side, and on the other side, it attaches to a set of membrane linking proteins that go through the sarcolemma and attach to the endomysium. Remember that endomysium is the connective tissue that surrounds the muscle fiber, and that endomysium will eventually blend together to with all of the other peri and epimysiums, and that will make the tendon. So this is the protein that connects the contracting sarcomere to the connective tissue surrounding the cell. Without this protein, your sarcomere can contract all at once, And that force will not be transferred to the cell, and the cell won't actually contract, or at least it won't actually contract very efficiently. And in people who have muscular dystrophy, which is a genetic disorder, this dystrophin protein is malformed. And so they have a problem with the working of their muscles. The working of their skeletal muscles is pretty bad, and that causes them a lot of trouble in their life. But for them, the real problem is going to be the fact that it also affects how the heart works. And over time, their heart is just not going to be able to stand up. You usually end up with accumulating amounts of scar tissue there. And we'll talk about that more when we talk about pathology. But dystrophin is going to be the protein that links the sarcomere and the thin filament to the endomysium and allows force to be transferred from the contracting proteins to the extracellular matrix. So the sarcomere itself is an important structure, and I wanted to sort of show you a picture of an actual sarcomere, right? So this is an electron myograph. As you can see, this is not just us like drawing pictures. This is an actual thing inside of the cell. This is an actual picture of a cell. You can see here, this is the surface of the cell. This is the sarcolemma. This is the, here we have the Z disc going down here. Here we have the thin filaments, and you can see that they look thin. Here we have the M line. The M line isn't easily visible with a microscope unless you highlight it, but the M line is there. And here we have the thick filaments. Note how they look thicker. And then this is our zone of overlap right here, where we have both thick and thin filaments. So remember that the A band is the entire length of the thick filament, whether or not it's overlapped. The H zone is just where you have just thick filaments, no thin filaments. The I band is where you have just thin filaments, no thick filaments. And we're going to talk about how these structures change when the muscle contracts. This is basically in its at-rest state. The way that muscle contracts is by what's called the sliding-filament mechanism. So this is what's happening when the muscle contracts, right? So here we have our actin in our thin filament, and here we have our myosin, our thick filaments of myosin. And we have our head groups, which is actual motor proteins here. So in this state, calcium is around, and so you can see actually calcium here has bound to the troponin, and the troponin has moved these tropomyosins out of the way, and that has exposed the handholds. And the handholds are these little black dots that you see on the yellow circles, right? So this actin is ready to be grabbed. Myosin starts off bound to ADP, and when it is bound to ADP and phosphorus, it can bind these handholds. Boom. Here in step 2, we've bound those handholds. When it binds the handholds, it lets go of the phosphorus. There's going to be a change at every step in what it's binding, right? So here it was binding ADP and phosphorus. It grabs hold of the handholds, it lets go of the phosphorus. All right, all well and good. ADP is then displaced with, ADP is then leaves. And when ADP leaves, the cell is now, or the filament is now charged. it causes the myosin cross bridges to rotate towards the center. So here we had, here the myosin, no, it's tilted like straight up. Here the myosin is rotated more in. We just moved a few microns in. So that was the pull. The pull is actually caused by ADP leaving. This is called the power stroke. So ADP unbinding from myosin causes the power stroke. The myosin now pulls. And now you're thinking, but I thought you said it burns ATP to create the energy to do this. And it does. But there's actually many different ways that ATP can be burned. And we're going to actually use every single one here, right? So when the phosphate leaves, that can cause something to happen. When the ADP leaves, that can cause something to happen. When ATP binds, that can cause something to happen. And when ATP is finally burned into ADP and phosphorus, that can cause something to happen as well. So with one ATP molecule, you can cause 4 steps to happen. Here the step is the ADP leaving. That's what causes the power stroke. Boom. But if you didn't have any ATP around, it would just say stuck. You'd get one power stroke and that would be it. You'd get no more contraction. Wouldn't do much of anything. When ATP binds to the myosin, that's what causes the myosin to let go. So here you see that the myosin has let go from the actin, and then when the ATP is hydrolyzed into ADP and phosphate, that causes these head groups to twist back. They're still stuck going off to the left here. They're still going off to the left here. Now they're going to reset when you hydrolyze the ATP to being pointed more up. So it's kind of like cocking a crossbow, right? So you think about a crossbow, you put the energy in when you pull the string back. But that energy isn't released until you pull the trigger. So in this case, this ATP being hydrolyzed and that changing the orientation of the head, that cocks the crossbow. Phosphorus leaves and you bind to actin. You're putting the arrow in the crossbow. When ADP leaves, you're pulling the trigger. And all of that energy that was stored in the orientation of those head groups is released, and the head groups move. You're pulling the trigger, sproing, you shot the arrow. ATP binds, and you put your cocking mechanism on, and then you use the energy of the ATP to cock your crossbow again. And that happens over and over and over and over, and that's what creates the force, the pulling force of myosin on the actin. All right, so now we're going to put all of that together and actually sort of watch what happens. So here we have a sarcomere, right? We've got our Z disc over here. We've got our thin filaments coming off from the Z disc. And these thin filaments have tropomyosin, that's this yellow squiggly here, and troponin on them. And currently the tropomyosin is blocking it, right? So currently nothing can bind to them. We have our thick filaments with our myosin head groups all facing out like that. And the thick filaments are all bound together with the M line, and they are attached to the Z disc by titin. Right? Up here, we have dystrophin, which is attaching our thin filament to the membrane. Note the size of the H zone. The I band, you can't actually see the entire I band. We're going to show half of the I band, the half of the I band from the Z disc to the zone of overlap. We have a zone of overlap down here, and the A band, which goes the entire length of the thick filament. We're actually going to be taking up from where we left off in the action potential lecture. So remember, with the action potential lecture, we had our action potential. It went down the sarcomere, or the sarcolemma, and it caused the release of calcium. So what has just happened to this muscle is that calcium has been released. So you can see that there's a whole bunch of calcium around here. And this calcium has just been released, and we're going to see how it causes muscle contraction. So the first thing that's going to happen is that the calcium binds to troponin. When calcium binds to troponin, the troponin moves tropomyosin out of the way. Now that tropomyosin has been moved out of the way, our myosin head groups can bind to the actin thin filament. So now we're going to undergo our sliding filament mechanism, and we have a power stroke. That power stroke pulls the thin filaments in towards each other, and see how that force has been transferred to the membrane, and the membrane has contracted just a little bit. Now, the rest of our cycle happens. We let go of the thin filament, and we reset our head groups. Then our head groups bind to the thin filament again. And then we have another power stroke. Now at this point, this muscle is fully contracted. Like, in reality, it's going to be a lot more than 2 power strokes to get a muscle fully contracted. But in this demo, you can tell that this muscle is basically fully contracted because these thin filaments are now running up against the M line, and the thick filaments are now running up against the Z disc. So this can't contract anymore because you actually have what's called steric hindrance. You're running into the Z disc, and you can't pull the Z disc past the thick filaments. It just, physics won't let you. Note what has happened to our zones. The H zone has shrunk dramatically. As the muscle contracts, the H zone shrinks, and the zone of overlap expands. The A band has stayed exactly the same because the size of the thick filament hasn't changed. So the A band never changes. As the muscle contracts, the H zone shrinks, the I band shrinks, and the zone of overlap expands. Now, at this point, what's happened is we're no longer getting action potentials, right? We got an action potential. It caused us to release calcium, and now we haven't gotten another action potential. Well, you have constant mechanisms that are scooping up calcium and sticking it back in to the sarcoplasmic reticulum. If you don't keep releasing the calcium gets scooped up and goes away. So we're going to disengage our thick filaments. And now the calcium gets scooped up and put back into the sarcoplasmic reticulum. And note what happened when the calcium left. Troponin put tropomyosin back into place. So these head groups here, this myosin, can no longer bind the actin. Right? The actin is now protected. And with the actin now protected, the titin is going to spring back out and cause the muscle to reassume its original shape. And that's muscle contraction. That's what's happening in your body every time your muscles contract. Now, there's a few important things to note here, and that is the length-tension relationship. So if you look at the previous slide, let's go back here, right? So when you're in this circumstance, some of your myosin head groups can bind your actin. If we were to stretch this muscle out, you would only have just a few bound. So when you stretch a muscle, you have less contact surface between actin and myosin. That makes the muscle weak. If a muscle is too stretched before it's stimulated, then you have minimal overlap between thick and thin filaments, and that means that you don't have very many head groups engaged, and your muscle is weak. If your muscle is overly shortened before being stimulated, then that means that you can't generate much force because the thick filaments just butt into the Z-disc and then they can't contract anymore. The long and short of this, see what I did there, is that your muscles are strongest when they are partially contracted. This is called the optimum resting length. So your muscles are strongest when they're not too stretched, but not too contracted. Your body wants to maintain your muscles in a position of optimum strength just in case you need to use them. So that if, somebody jumps in through your door with a chainsaw and a weird mask, your muscles are in their position of optimum strength to, throw your glass of wine at them and jump out the window. Or something like that, however it is that you react. Your brain might not do a very good job of it, but your muscles are going to be ready, gosh darn it. The way that your nervous system maintains your muscles in this state of optimum readiness is that it maintains what's called muscle tone. Muscle tone is a state of partial contraction. Basically, even when you're not actively intending to use your muscles, like I'm not thinking, I'm going to be moving my arm or I'm going to be moving my legs, my brain will still send occasional impulses. Bidip, 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 down to those muscles, even though I'm not actively using them, to cause little micro contractions and just keep them in this state of being partially contracted. Stop them from becoming too relaxed. The state of being too relaxed where there's no muscle tone. The muscle is completely uncontracted. That's called flaccidity, or being flaccid. Flaccid muscles are weak. So here we see an overstretched muscle. And It is weak because it has very little zone of overlap, and it's only in the zone of overlap that force can be exerted. Here we see an overly contracted muscle. It's weak because basically it's already as contracted as it can get. There's no real room for more contraction to occur before the thick filament starts bumping up against the Z-disc. So it just can't get a real run of things. This is the state of optimum length, this partially contracted, where we have the most contact between our actin thin filaments and our myosin head groups. This muscle is in its strongest state. It has a maximum zone of overlap, but it still has room to contract. So in summary, what you need to know for this section: you should know the steps and events in muscle contraction, and the sliding filament mechanism. So you should know those 4 steps of the sliding filament mechanism. You should know the role that ATP is playing. You need to know how the calcium troponin tropomyosin system works. When calcium is present, troponin moves tropomyosin off the actin binding groups, allowing myosin to bind to actin. I know that there's a whole bunch of complicated words here. Use them repeatedly, write them down, you'll eventually remember them. Understand the role of the supplemental proteins: dystrophin, titin, myomycin, and nebulin. To reiterate that, dystrophin is what's going to conduct force from the contracting sarcomere to the membrane and the extracellular matrix. Titin attaches the thick filament to the Z-disc. Myomycin is the protein that makes up the M line, and nebulin attaches the thin filaments to the Z-disc. You need to be able to draw a sarcomere and label all the pieces. Like, I'm not saying that that's going to be on the test, but I'm saying that it might be on the test. So you should be able to sit down there and draw a sarcomere with a Z-disc on either side, thin filaments coming out, thick filaments in the middle, be able to label the Z-disc, the thick and thin filaments, the H zone, the I band, the A band, the M line, and you should be able to show how the sarcomere changes in different contraction states. So if I say, draw me a sarcomere that it is fully contracted. Show me a sarcomere that has been fully stretched out. You should be able to know what those means and draw it under those conditions. Last, you need to understand the relationship of stress and contraction to the ability of the muscle to exert force. Understand what muscle tone is, and how a partially contracted muscle is a muscle that is capable of exerting the most force possible. I know these were a complicated set of 2 lectures. We're going to go over some of this information in class, but you may want to watch these videos a couple of times and sketch out the process of what's going on.